welcome to a special episode of Dairy Digressions, the new podcast of the American Dairy Science Association, the Journal of Dairy Science, and JDS Communications. I'm Matt Lucy, Editor-in-Chief of JDS Communications, and I'll be your host. I am coming to you from the 2023 annual meeting of the ADSA in Ottawa, Canada. This global gathering of dairy scientists convenes every year and brings together the top minds in the dairy industry. For these special episodes, I'll be sitting down with experts who are attending the meeting to get their thoughts on the meeting, the sessions, and the exciting new developments in dairy. If you enjoy Dairy Digressions or have any feedback for us, please let us know at adsa@adsa.org. Make sure to like, subscribe, and rate us on whatever platform you're listening on, and spread the word to a friend or colleague. Now let's get to the meeting. Today, we have with us Dr. Jeffrey Firkins. Jeff, welcome.
Thank you.
We have a nutritionist in the house, okay? And of course, the last one we did, we had a repro guy in the house. So it was a little trash talk about nutrition and genetic, and the repro guy was trash talking. I would never trash talk, would I?
No, and
No.
he should have learned you don't do that when the next guy after him gets the
Gets to say it right. Last word wins. So Dr. Firkins is a professor in the Department of Animal Science at the Ohio State University. He was raised on a grain and livestock farm in northern Illinois and developed his interest in dairy cattle by working on his uncle's nearby dairy farm. How many cows was that? How many were you milking back then?
Yeah, they only had about, I guess, about 50 cows.
50?
Yeah, I remember actually pouring milk into a bulk tank.
Oh, yeah.
You could still do that.
Old, probably, were you using the old Surge bucket milkers or something like that?
Yeah.
And did you feed the cows by hand?
Oh, yeah. It was my uncle and cousins that mostly did it. I was mostly there as, conscripted labor.
Get the Firkins kid over here. Well, that was your uncle. So and then Dr. Firkins has his BS from Illinois, MS Illinois, and a PhD, and also did his postdoc there in dairy nutrition. And as we've talked, Jeff is his mentor was Dr. Jimmy Clark, famous dairy nutritionist, long time member of ADSA and president, I think, at one point as well. And we wish him well. Jimmy, if you're out there, we remember you. All right. Yeah, we remember you.
And I did PhD with Larry Berger there.
Okay. So
yeah, I did postdoc with Jimmy.
Okay. So
and everybody who postdoced with Jimmy got a good job.
So then you moved to the and then after that you moved to Ohio State, and then you reached the rank of professor in 2000. And of course, Dr. Firkins is just well known for his work as a dairy nutritionist. He lists his three main areas of interest in the interface between nutrition and microbiology to enhance the conversion of protein to microbial protein, obviously essential for what cows do, the formation of microbial protein, interactions of the microbiology and fiber and starch digestion and biohydrogenation, other sort of essential features, and then improving the quantitative predictive of protein and carbohydrate digestion and microbial protein production in dairy. So really a true rumen kind of microbial nutrition guy. You've probably seen it all. You've probably seen it from, I'm just thinking, from your days as a grad student. There wasn't a lot of DNA sequencing back there, was there?
No, they did know what DNA was.
We did. Well, it was after Watson and Crick.
Yeah,
okay, so we had that after. Paper. You probably cited it in your dissertation. But then the 16S sequencing, we were talking earlier. Okay, so now, as you know, as you're very well aware, we started out. Okay, I'm an old guy too. Okay, so I got it. All right. So, but the 16S sequencing, you were mentioning that you were right at the start when people started doing 16S sequencing.
Yeah, a lot of that was really developed at the University of Illinois. There was a guy there named Carl Woese who got a lot of attention. I was really lucky. I was so interested in microbiology that I decided to go and do a PhD instead of go to vet school.
Oh yeah, okay.
So it's like, I wanted to work with ruminants
yeah,
and just to find out what all those microbes were doing. Back then, there was about 50 species, maybe.
we
Now-
wish,
Yeah.
yeah, right.
Yeah,
Yeah.
right.
It's
So-
those things changed then.
Yeah, they did a lot. And in fact, like I remember listening to the first time they
Yeah.
did that. The first work they did that on that was actually with RNA. And then,
Okay.
yeah, and then PCR came out after that.
Yeah.
And pretty soon everything went that way.
Yeah.
But they have a tremendous microbiology program at Illinois. And
Yeah.
so I took all, as far as I know, all kind of the same classes that the
Yeah.
microbiology students did.
I think one of
Hung
the
around
things,
with them, they let me, they included me
yeah,
like the little brother.
well, and I think one of the things to emphasize is people don't always talk about Illinois for its great I don't know why not, for its great scientific basic and applied scientific achievements, but it's honestly, it's an outstanding institution. It's a big institution, a lot of fantastic scientists there doing really great work, and I think I don't know why it gets forgotten, maybe because it doesn't toot its horn hard enough, or maybe I don't know why that is, but there's a lot of good work at Illinois.
It's very multidisciplinary. I mean, like I was involved in the interdisciplinary program in nutrition at
Yeah.
Ohio State,
Yeah,
but they had the same thing at Illinois. So interacting with human nutrition people, I had a few people who were as professors who were in the National Academy,
yeah,
and you just got this general idea of how nutrition fit in, including with repro.
yeah, including repro, and of course, the dairy is still Illinois. It's still. I remember this is a digression, but you used to have that 5-breed thing going on at the dairy. I don't know if that's still going on at the dairy, if they got all those different breeds there or what.
I'm not sure. There at LSU, we had 5 breeds when I first started.
Is that
5.
right?
And yeah, and they had different size stalls for every breed. It was that was kind of cumbersome, and of course, then we moved down to 2, and
Yeah.
then the poor Jerseys, they were either swimming in stalls or there weren't very many for them. So
so you must, and I will get back to the program in a second, but being a guy, okay, who saw as, again, earliest phases of, okay, so I don't know what happened. I don't know how rumen microbiology used to work in the beginning. Did you take a sample and plate it out and try to figure out what bacteria were there based on cell culture or microbial culture?
yeah.
Was that the way? I mean, you're like, boom, what's here?
Yeah, I mean, when we all took, if you were in ruminant nutrition, you took this class, and Marv Bryant, who was in the National Academy, taught you how to do this.
Is that right?
Yeah, they had the Hungate technique named after the father of Rumen Microbiology, they called him then.
Yeah.
And so you would take a tube, and you really needed 3 hands to do it, but you learned how to hold the tube in your stomach and use your stomach as a hand, basically, and you uncork the tube with your thumb and your first finger there, and then do your other stuff there.
And it
like
was the way you did it, and you'd have to,
Yeah.
and then we got 16S sequencing, and of course, and then now it's just all metagenomics and all whole genome sequencing, all this other stuff. And as a rumen microbiologist, okay, so probably 10, 15 years ago, they started talking about the microbiome, and everybody got into I mean, even Matt Lucy does microbiome stuff now. I send off sequences and I analyze I mean, I do microbiome, okay? And you must have been like, what? Like, we've been doing microbiome forever. No one even cared. They wouldn't fund our grants. And now all of a sudden they can't throw enough money at microbiome.
No, you just need a sample.
You just need a sample, and you run it through some program, completely changed.
Yeah. Yeah, it's all changed. And of course, at OSU where I've been fortunate to work with some really good microbiology people.
Yeah.
Burke Doherty was one of a handful of people who could look at protozoa and identify them, and his word was law,
Yeah.
He said it was that it was, and so on. And Mark Morrison was there for a while. He was a great colleague, and ZT, who I've worked with basically the whole time he's been there.
Yeah.
And ZT really was at the very forefront. I remember when he interviewed Mark Morrison, goes, "What'd you think?" And I said, "I liked him. I thought he was good." And
Mhm.
Mark So I said, he can isolate RNA and DNA.
Mm-hmm.
Why is that such a big thing? Because I
Mhm.
didn't really know. And he goes, because he can, mate.
Yeah, yeah, right. Because he
back
can
then,
do that, right?
It was to really be able to do that well. He was, I mean, there was just very few people doing it.
Yeah.
The technique that's widely used by everybody, anybody who takes samples from the rumen or feces or
Yeah.
whatever, you and Morrison,
That was
they,
the technique.
yeah, they knocked it out, and it's been cited. I don't know. It's too bad it wasn't published in JDS. It definitely helped our impact back
Yeah,
there.
yeah, these yeah, they still bring up that old Pete Van Soest paper, that's the one, right?
Yeah.
And I don't know if Pete's still alive. Is he
No, he's
deceased?
not.
He's
He
deceased.
passed.
Okay,
Yeah.
but just classic, right? And so so here we go, microbiome. Everybody does microbiome. Now, is it still true that the majority of species, okay, I know you're a protozoa guy, and I know protozoa are not bacteria, but so we'll leave protozoa off to the side for now. But is it still true that the majority of bacteria in the rumen cannot be cultured?
Yeah.
Why
Yeah.
can't you culture them? Just throw them in some anaerobic stuff, and why won't they grow?
Well, they'll live all together in a culture, but when you got to turn them into a single culture, you got to start with one. And
Yeah.
so that means you got to disperse them out. And the standard way to do that is with agar.
Yeah,
And some of them just can't live with agar.
yeah.
So
They can't live alone. They need their friends, and
some of them can't live alone, and some just can't live in agar.
yeah,
But some, people have found ways to culture different kinds. So
yeah,
the cultures are becoming more complete. At least
Yeah.
they're close enough that you can, like, there have been some taxa that even all the way to the phylum level, there's no cultured representative, but
at the phylum level.
that they're closing in on trying to figure out how to get some of those
Yeah,
done. So there's lots of reasons. Some of it could be just they didn't let them go long enough.
yeah, yeah. So you mentioned early in our interview, you said about 50 species kind of when you started, and based on the latest metagenomic sequencing, what kind of numbers are we looking at in the rumen?
Yeah, it depends on what you call a species, which
Yeah.
is being
Mhm.
questioned, because it's all based on whatever the cutoff to group some into one category
Mhm.
and not another.
Mhm.
But my colleagues did a survey of all the ones that were found, and there was, I don't remember, 6,000 or 7,000.
Oh my gosh, 6,000 or 7,000.
But you don't usually see that in a rumen.
Yeah.
It's more like, maybe 1,000, or
Yeah.
depending on how where it came from, you can have maybe more.
Yeah. Okay, so this is a bit of a digression. This is something I've been thinking about. Okay, post-rumen fermentation. Okay, we always think large intestine, but that's my interpretation, dummy repro guy, large intestine. We just, the purpose of the large intestine for us is just, put our hand in it. That's all we need it for, right? Repro guys, but you nutrition guys care,
We
okay?
still put our hand in it.
Yeah, right. I'm always like, we need that large intestine because we got to get to the uterus, okay? But
For us, if it falls on the floor and you know whose it is,
Yeah,
it's still good enough.
it's still good. But so where does post-gastric fermentation start? Is it solely in the large intestine, or do we get post-gastric fermentation in the small intestine?
There's some in the small intestine,
Right.
sure. Before the end of the small intestine, pH starts rising enough, and so on, and they start appearing. They're just not really, really growing so much to really break down significant feed until really after the small intestine.
Okay.
So
Does it matter what's going on in the large intestine, or is that just sort of it's a not much in terms of the total nutrition of the cow?
well, we look at site of breakdown of fiber and so on. Some of it occurs there.
Yeah.
And so it might be 10-20% of the digestible fiber occurs there, like that. So it does matter,
Mhm.
like some people look at the large intestine in part because of inflammation that animals have. And if they have acidosis in the rumen, you're likely to get some down there. And then you probably know this, but the structure of the large intestinal wall is more simple than in the rumen. The rumen knows there's supposed to be all these microbes trying to do things. So there's 4 layers, but in the large intestine, there's only one.
Yeah.
mucus is in there, and we know that if animals get acidosis, then you start seeing mucin casts and so on. But yeah, I think it matters, and
Mhm.
it might be for reasons not just so much about how much fiber is broken down there. Any microbial protein is really past the absorptive side, so that kind of doesn't matter. But with respect to the whole animal's health and so on, yeah, it matters.
Yeah, and that's this leaky gut concept. That's large intestine. When they say leaky gut, is that
Yeah.
large intestine they're talking about?
They're referring to it there, but you can have leaky rumen.
Yes.
Yeah. So it happens, and
Yeah.
there's some people studying the rumen wall and its role.
Yeah.
I think it's more important. I think we need to be doing more in it.
Yeah.
In that area and capturing samples from there.
Yeah. So I'm going to continue on. I kind of didn't finish your bio here, but just to remind everybody that we know each other through our work on the journal. I mean, you are currently you're probably senior section editor for Dairy Nutrition right now. You've probably been at it for about 6 years now. I don't know if this is your second tour of duty, your third tour of duty, or
this is my 6th year with JDS.
Yeah.
But you're probably senior for 4 years. This is my 4th year.
And so I don't know if I recruited you or somebody else recruited you. I can't remember, but so we've known each other for a long time through the Journal of Dairy Science. And then it mentioned here that you're also involved in the USDA competitive grant system in a variety of ways. And of course, you're very active in the national, international ruminant, very hefty publication record, very sought after, invited speaker, lots of PhDs and MS students, and you also teach. You teach nutrition. You mentioned you taught carbohydrate metabolism. You teach in classes in addition to carbohydrate metabolism.
Yeah, so I teach, PhD level carbohydrate and amino acid metabolism, and that's really fun because I get to keep up with that. Most of the students are human nutrition.
Oh, wow.
Yeah. So teaching them that microbiology means something. And of course it does for humans too.
Yeah.
And amino acid metabolism is important, even if they think it's boring. Right?
Yeah. Yeah. I was interested in that last speaker. I remember she was talking about fecal transplantation, and how that, I remember when I first read about that in Science Magazine, they were talking about treating colitis. I think it was colitis to treat with fecal transplantation.
Not just colitis, but
other diseases as well.
So
and this
happens.
This was doctor doing it. And they were like, this science didn't think he was crazy. And this guy didn't think he was crazy. And look where we are today, that people really believe that if you try to move the microbiome in one direction or another, it can have significant health consequences, good and bad if it goes in the wrong direction.
Yeah, there's so many. That's the nice thing about like teaching carbohydrate metabolism for a lot of human nutrition students. Fiber is important. When I was at Illinois, I interacted with George Fahey, who was one of the experts on fiber. Yeah. And there's all kinds of things that you can lay the groundwork for us not getting enough fiber as being really important. Yeah. So I want to just mention that you are a fellow of the American Dairy Science Association. That award was made in 2020. And of course, that's one of our highest awards we can bestow. And it just recognizes you for a lifelong research and service. And that's congratulations to you. American Feed Industry Award from ADSA, Applied Nutrition Award from ADSA. So you've got your share of hardware, right? You got your share of hardware. You've done well, my friend. You've done well. Thank you.
So tell me what's going on in Ohio. You're from Ohio. Ohio is, we don't put it at the top of our dairy states, but we don't put it at the bottom. Where are you at? I don't even know where it ranks.
I think we're 11th.
11. Okay, not bad. Not in the top 10, but dropping fast because all these other South Dakota and everywhere else is pushing their way. Idaho. I'd go Idaho.
They took us over.
They took over. New York. New York's dropping too. So you're in good company. Okay. But so what's going on in the local dairy scene in Ohio right now? What are the farmers talking about? What are they concerned about?
Well, they're, of course, always interested in making more money and milk price and so on. And milk price has gone down a bit.
Yeah.
So that in Ohio, we have several fairly medium-sized cities,
Mhm.
so there's a rural urban interface. I always have a hard time saying that.
Mhm.
And so we have some issues related to people living nearby. We have waterways. We have a lot of interest in trying to decrease the runoff of nutrients into
Mhm.
those waterways.
Mhm.
We've had a few issues with algae blooms.
In Ohio, in the Ohio River, or where are these algae blooms?
In Western Ohio, and then so there's a big pocket of dairy in Western Ohio.
Oh, yeah,
They
yeah.
have a one lake there, and they just happen to have one of those algae blooms that's not good for
Mhm.
people.
Yeah, for sure.
And so it's an issue that people are really trying to decrease. So we, like everybody, are having fewer farms, but larger farms. And for a while, a lot of the farms were like going to Michigan. We hate
Yeah.
to say, we at Ohio State, we have to have M with a crossed out when we play Michigan.
Michigan. Oh, ouch, right?
Yeah, especially the last couple of years. I say we've turned from the Ohio State to the Ohio State when they beat us.
Yeah, yeah.
But yeah, but we're having expansions as well. And
Mhm.
so actually they've been doing pretty well in the last few years with
Yeah.
ranking of states. So when I started at OSU, we were 7th.
Yeah.
Yeah. So we've been like 10th, 11th for a number of years now. So
Yeah.
yeah, there's a vibrant industry and
Yeah.
some good leaders. We just went through an interview process, and we had a couple of really outstanding members of the Red Dairy Farms who were participating, and dairy farms or dairy nutritionists in the field. So we have a good dairy industry, and
Yeah.
they need more students to come and work.
Yeah.
And so it's something we're trying at OSU to help train students to be a little more accomplished and be able to go into that instead of something else. So
I think the South Fork Dairy in Ohio, South Fork Dairy, maybe. It seems like I spoke with some Ohio dairy producers, and they were really sharp. They were smart guys.
there's some people there that are as good as anywhere.
Yeah,
I mean, they
smart
really
guys. Really. I was like, whoa, these guys, they know stuff. And I think you would agree, Jeff, that's one of the things that Paul and I talk about and we think about. It's like when you go talk to these really good dairymen, they know stuff. They know a lot of stuff. They've listened to your talks, and they understand what you're trying to tell them, and they ask good questions. And I'm always like, whoa, these guys are switched on, and
something I always have figured out when I go and talk to people in applied venues like that. I don't try to take the place of good people in the industry, because when I started, maybe they weren't at the top of their game like they are now, but there's nothing I'm going to say that they don't already know on an applied standpoint. I try to explain modes of action and be there to help reason things and offer like that. And so I feel like they want to know if something's new, does it work? When would it work? They want to ask questions. I remember one time I was given I've done several of these sort of like workshops, kind of where they'll just bring in, they just want to ask questions. And I said one time, I'm not really sure I helped you at all. And the guy goes, whenever we talk with you, it always helps us to think better. We go back to our roots, and because we're locked in on doing the same,
right.
We're trying to get everything done in the day. They're all busy. You help us to rethink. And I feel like that's the role of academics with people in industry, is to help in that venue. A lot of good people, and they're just trying to find out more about more things. So.
Yeah, they just are just it's always you might seem a little weird, but that's not the right word, weird, but it might seem, maybe people don't understand. But when I give a talk, I am most stressed when I'm talking to dairymen.
Yeah, me too.
They are, because I can talk to academic, and that's one thing, but you talk to dairymen, And there's stuff you just don't know about their world, and you bump right into it, and then you go, oh, okay. I remember I was speaking to these guys from Ohio, and they wanted me to talk about transition cows, and I was talking all about transition cows and transition cow pens and everything. And I knew I was telling them stuff they already knew, but one of the guys stood up and says, "You don't understand." He says, "You're talking about my transition cow pen is that every month I have 300 cows in my transition cow pen." And I said, he goes, "That would be nice, but there's some months I have 500 cows in my transition cow pen because of the way the calves are coming in. And there's other months I got 100, and I'm not going to, and that's the reality." And I thought to myself, sometimes the reality doesn't fit the recommendation, but it's still the reality. Does that make any sense?
Oh, yeah, I think so. And, every place is different. And that's what's kind of fun to interact with people is it's never, I mean, every dairy's got somewhat different things, even if no matter what they're doing.
Yeah.
And, what you said about, well, in this case, it was, transition cows, but there's a lot of aspects that they know what they're doing and what they want. And I think they even know what we would tell them is the perfect world. They're just trying to figure out how can I do the best I can under this situation. So,
yeah. So the American Dairy Science Association, this meeting, I'm sure you've been coming here forever. And that's, I don't know. What does your tag say? 40 years, 5 years?
Almost 40.
Almost 40.
I asked Don Palmquist. I think I spilled something on my tag. Oh no, that's a star.
So you've been here forever. I think
it's been almost 40. I think the first one might have been in 84 when it was. I asked Don Palmquist, when was it at the University of Illinois? That's the first one I went to.
You went to, yeah. So the thing about the dairy nutrition community, they support this meeting. I would say they come, no question about it. Okay. So I guess the question I'll put to you is, why do they come? Okay. So I will just tell you, I was walking through, if there's a room and there's 20 people standing outside trying to get in, it's usually the nutrition. They come.
I don't totally know, but I think some of it's they come to interact, of course, and to find out new things. I think they like to go and talk to a lot of other people. I've been really surprised at, even with different companies and competition and so on, they come and just want to go and talk about common things and how's that doing and interact. And, like if they're always hearing about something new. And what is it behind this? They don't really have time to go and read all the literature like you and I do. We have all kinds of time to do that. Right?
We just sit around reading books. But
they don't have time to do that. They want to come, and they all usually have something in particular, but they want to find out more about it. And if they don't know, they get on the phone. One person told me one time that I was more well-known outside of Ohio than inside of Ohio.
That's probably true.
Because they said if it's something about this subject, they look up who's somebody to talk to about that, and they call. Of course, I was fortunate to have Bill Weiss as a colleague, so they always called him first anyway, if they were in Ohio.
Now, you don't have an extension appointment, or do you?
No, I don't.
Yeah. Did Bill have an extension appointment?
Yeah.
He's just retired, and I know he did have an extension. And would you say, okay,
and Maurice Eastridge, who are both excellent. So
do they? And I know there's a Bill Weiss symposium at the meeting, and of course we all I know him. I don't know if he's retiring. Is that why
he's retired? Yeah, he retired.
And then, guy like that's still going to be active, probably consulting or working with people, I would imagine. But what would you, we're going to get to the focus topic here in just a second, but what would you, if you were to say Bill Weiss, people know him as a mineral nutritionist and vitamins and minerals, or they know him as something else than that?
Well, that's interesting, because in the symposium for him, there's one on energy, one speaker on energy, Paul Kononov.
Oh yeah.
One on vitamins and minerals. And the other one on I've forgotten.
Okay, well, it's okay.
Amino acids.
Yeah.
Even did work on that.
Yeah.
Protein. So Bill did a lot of things when he started. He was really known for forage.
Is that right?
And there's not one on that, but there could have been,
Yeah.
but he's
Yeah.
really well known for energy. But yeah, vitamins and minerals is the one. I think it's not just because he did
Yeah.
a lot.
Yeah.
There's no Bill Weiss sitting in the wings to take over that void.
That, yeah.
I mean, I don't mean any disrespect to anybody
Yeah.
who's doing that. It's just on so many vitamins and so many minerals.
Mm-hmm. Mm-hmm.
He was really like E.F. Hutton. You remember
Yeah.
that
Yeah,
commercial?
for sure.
We probably got to edit out.
Yeah.
somebody.
Yeah.
But he was like that when he said something, it's like that's what they came to see.
Yeah.
If he said do this, everybody did it.
Yeah.
Honestly, I heard many talks with him saying, well, we need to decrease the amount of this mineral and here's why. And then a bunch of people went and did it.
Yeah. I wish I could put that personality in a bottle. I mean, he does have a unique personality. I'm not saying bad. I'm just saying that guy, when there's something about that laugh and that look he would give you, and
Yeah.
I don't know, just that personality was just great.
Yeah, he's very when he gives a presentation, he's very this like that. But when you get him outside of it, he's really funny.
Yeah, I believe it. Yeah, so so that is going to be so that's I know that's a session you're looking forward to. That's going to be a little later in the meeting.
If he gets here.
Oh, is there a problem with the flights and stuff or what?
Yeah, he's still in Newark.
Oh, no. Oh, Newark. Ow. Well, he needs to get here. And I think that's tomorrow or maybe the next day. Wednesday. Okay. So so I want to ask you about, I know one of the things that's on everybody's mind globally, okay, is methane production from ruminants. And in our last Dairy Digressions podcast, I talked about the fact that there was a paper published in PNAS, and it just said that, well, if you look at mammalian biomass, okay, of mammals, people are there's more biomass in people than anything else, okay? But then ruminants, domestic ruminants, are equal to people in terms of biomass, and I thought to myself, how could that be? Are there that many ruminants? But you think about America, there's 300 million people in America. If you add dairy cows and beef cows, there's probably 40 million dairy plus beef, probably somewhere in there. And cows are bigger than people, right? So it doesn't, maybe a cow is 5 times as big, and you get pretty close to the same amount of biomass. Okay, so you have a tremendous amount of biomass,
Yeah.
And that biomass produces methane. And I know we can argue it any way you want that it's methane, it doesn't matter. It's the CO2 that matters. It's the nitrogen, it's whatever, but methane is on people's minds, as is CO2. And methane, as you know, there's 2 sources. There's enteric methane that's either going to come from the rumen or from the rectum. And then there is also methane from manure piles. And of course, that's the source of biogas and capture that. But as you also know, a farm's got to be a certain size before those digesters they work. And I think you got to be. I was listening to one of your dairymen. I think he was talking about his digester, and if you're at 1,000 cows, I don't think it's really enough. I think you got to be 3,000 or something. You got to have a lot of cows to make those digesters work.
Yeah, I'm not totally familiar with how big it needs to be to really work, but yeah, that's good way. And but there's other ways we can do it. Like we're doing some work with this biochar. You can collect feces and turn it into biochar, and biochar is active.
Okay, now you have to slow up and tell me about biochar. Okay, so now we're talking methane. Now we're talking methane that's from manure. Okay, and that's called, I don't know. It's from the manure pile. I don't know. It's not enteric. It's the stuff that comes off the manure pile. And again, so if you just take your manure and put it in a pile, it's going to emit methane for whatever reason. I don't know. I guess there's bacteria or protozoa or something that makes methane. Yeah. And then I said, well, you can try to capture that, but that's expensive. Now this biochar, I have heard about biochar, but what is it?
Well, there's more people looking into it, and there's lots of scrap biomass that you can just let decompose on its own, and then it's going to produce CO2 anyway. It's going to break down and so on. Or you can sort of partially char it, and then you have this product that does a lot of things. So some people are looking into that as like a soil amendment and so on. And there's some potential that it does some things in the rumen as well. So we're doing some research on that now.
So you char, do you burn it to char it? When you say biochar, do you burn it, or how do you make biochar?
It looks kind of like charcoal, but it's not, it's fine.
And then you put that into the soil, and that increases the organic matter in the soil.
It's got a lot of organic matter in it, but it's there's like lots of sites for minerals to bind to and so on. So for example, some people are applying it because nitrate binds to it, and then it keeps it from leaching out into the groundwater. So we don't want nitrogen leaching out. So there's lots of things like that people are looking at down the pipeline
to try to manage their manure better, instead of just, we used to, as you know, we used to just take the manure and put it in a manure spreader and fling it into the air and dump it on the top of the field. And of course, now you might knife it in. You'd be more likely to make a slurry and knife it in. At least that's not flinging it in the air, and that'll keep it from going somewhere else. And it sounds like this is another technique that might be able to be used.
Yeah. So my colleague at OSU is studying how to, you lose nitrogen during storage in manure, and we'd rather keep it. Then you have a better nitrogen to phosphorus ratio of the manure. Then when you apply it and you're burning fuel to apply it, you're applying more nitrogen too.
Yeah. So it's interesting how we really evolved, even in terms of manure. Manure, again, I'm not
There's
being
the science to it.
There's the science to it, right? There's the science to it, and drag lines and pumps and all this other stuff. And what's so funny, what's not funny, but what's interesting about dairymen is if you look away for 10 years and all of a sudden there's all this new technology that you don't know about that everybody's already using. They don't wait on technology typically, I think. And manure is a good one. I mean, these drag lines and knife and everything in, and custom and all that other stuff.
Yeah, I agree with you. There's lots of, the engineers are doing that. And then, the scientists are trying to find better, like I said, better ways to retain more of the nitrogen.
Yeah. And it's kind of interesting. We used to, you talk to certain dairymen. I don't know if it's still true, but it seems like, you talk to large dairies and that's on their list of things they need to, that they concerned about, they're worried about, is what am I going to do with all this manure?
Yeah. There's that, and not just the nutrients in it and when you can apply it, and phosphorus, and all that. If like in Ohio, there's, people comment when you do put it in, even if you knife it, it's still not odorless really. So there's I guess a real science of odor as well.
So the enteric methane, that's what I know it's an interest of yours, and how do you change the microbiology, the rumen? Maybe you're not gonna, I don't think you'll ever fully eliminate methane production out of the rumen. There's probably some biochemistry there that says you're going to be hard put not to have any methane. But so of your looking into your crystal ball, okay, what do you see as, what's the potential? Okay, so what's the future look like? What's the most likely approach? So we can throw some different chemicals out there. We've talked about monensin is a rumen modifier, 3-NOP, seaweed. Is anything possible? Can we actually reduce the amount of methane coming out of the rumen of a dairy cow?
Yes. So let me start by saying you're never going to get rid of it, because those microbes are essential. The fermentative microbes that feed them, they need to dispose of hydrogen, which is the main precursor for methane. And so you're never going to get rid of them. The big thing is,
so there's a biochemical pathway that has to be met. You're making hydrogen, H plus,
H2.
hydrogen gas, that's explosive, I thought.
Well,
Just checking. I'm just checking my chemistry here. So they make hydrogen.
they make hydrogen, and then the methanogens take up hydrogen, and that's
Okay.
they turn that into methane.
Okay.
Now there's some other microbes that can take up hydrogen too. And so people are looking at what can we do to help them out so they outcompete methanogens and don't make methane. So there's a role there, making some progress on that. To me, we need to start for methane. We always have to look at, we're never going to get rid of it. What we're trying to do is make the amount of methane be as efficient as possible relative to the amount of milk.
Yeah.
Right?
Okay. This is excellent. Right.
Yeah. So we have to improve management in all kinds of ways, forage quality and all these things, including repro. Repro has a better repro means fewer replacements, right?
Yeah. Right.
Or more of them that are beef, and grow faster. All that helps to decrease the methane load that gets charged against the amount of milk.
Right. Yeah. So this is an important point, I think. And I think, Jeff and I and many in the dairy industry understand that is everybody would like to go back to the good old days, but today's dairy cow is phenomenally more environmentally friendly than the cows of past, of our grandfathers, because they make a lot of milk. What you're saying is they make a lot of milk per unit of methane, essentially.
Exactly.
And so they make a lot of food. And so if you can, it's like the old argument, we used to learn, I don't know what you learned, but I learned, at Cornell was like, okay, I would rather have this one cow producing X amount of milk than 3 cows producing X amount of milk, because the one cow, you reduce your maintenance requirements. And is that still true? I mean, one cow is better than 3 in terms of environmental footprint. I think that's where we're at.
Yeah, sure. And the more milk any individual animal makes, the more of the energy she consumes and more of the methane produced is being paid for by more milk. So you're basically diluting out the maintenance or the fixed costs of it, if you will.
Yeah.
So that's all happening. Anything to do with improving that. When I was on this FAO project, Alex Ristoff was the leader of that at Penn State. And there's some really good information in there that explains all these managerial approaches that people can do to improve methane. That got a lot of attention. I worked with Joanne Knapp who authored some work on that. She worked with DMI, so on. And so there's all kinds of things there. With enteric methane, which you brought up, we have ways that we can do that. The first thing is better quality diets. If we get animals to break down feed better, there's again less methane per amount of feed that we give them. If we feed more grain, there's naturally going to be less methane. And it's interesting that if you look at enteric methane per ruminants and you look at the US versus other countries, we're way ahead of them, because we make so much more milk. We're feeding better quality forage. So there's much less methane produced per unit of forage. And so I would say we're not really the problem, but we can be part of the solution.
Yeah, fair enough. Right. And so I guess that sort of bumps into the whole concept of how much it's interesting how much milk we've had this, people have this discussion all the time, when do they stop making more? When does a dairy cow reach a plateau in terms of her capacity to make milk? It doesn't seem like that curve is tilting over at this point.
Yeah,
I mean,
just keeps going.
Isn't that
genetics
funny?
It's amazing.
It's amazing. Every year the cows get better. I don't necessarily, and we would both agree, I think, they're making more milk. I think they're just as healthy as ever. You can't make milk with a cow that's not healthy. Okay, we know that. I mean, sick cows don't make milk. Healthy cows can make milk, right? And it seems like, you can put together this package that just keeps doing better. Now, I always felt we used to always rely on just bigger and bigger cows, but now we got negative weighting on size, which makes them more efficient. In other words, we select for a smaller cow that makes the same amount of milk, smaller cow, less cells, less maintenance requirements, less nutrient requirements for walking around the barn.
We're selecting for animals that can eat a lot,
Yeah, they can
process it very efficiently.
Yeah.
You know, how well do they process when they ruminate?
Mhm.
You know, I mean, rumination is a tremendous gift to ruminants to be able to help break down fiber.
Yeah, yeah.
And then if we feed less fiber, then rumination is of course what buffers the rumen. So, you know, we really need animals that are really efficient at that.
Yeah. we're gonna wrap up in a couple of minutes, but I'm gonna ask you a question that I'm wondering about. You just mentioned eating and be able to eat. Cows, you know, we know all about time budgets, and I tell you what, we're a lot smarter about that now. You know, cows got to go to the parlor 3 times. They gotta spend a lot of time lying down. They gotta spend time eating, and that's another episode. But is there anything going on? Do you see anything happening in barn design in the future to accommodate these cows that in the lifestyle they need to lead? You know, I mean, you know, because is there anything happening, or are we just going to be freestalls forever? I mean, what do you think?
That's a question I'm not quite sure I'm capable of answering.
I should ask Trevor DeVries.
Yeah,
you know, get Trevor in here.
Yeah. I mean, I feel like there's better and better attention to cow comfort all the time and how they design things. So there is definitely some improvement in free stalls and so on. So I think that throughput, making sure animals have a place that's cooler, you know, better job with fans and all that, and water, good water. And so I think we keep making improvements because we need to, and there's a push to do it. And you know, it's always going to catch up there.
Yeah. So I'm gonna okay. I just got so many questions for you, but I'm gonna interject another quick one, and then we're gonna have to wrap up. All right, you guys got to turn the cassette tape over soon. So you remember those cassette tapes?
I remember 8-track.
Okay, so did Fricky. Okay, that's funny. So pretty soon we're gonna turn the cassette tape over, and Jess is gonna look at us and go, "Just a second." You hit stop, flip it over, start. Okay, start again, guys. You know, this is the and you said you're gonna put in a robot at Ohio State. And of course, when I've had calls when I was editor-in-chief at JDS, you know, from nutritionists saying, "Hey, my dean wants to put in a robot. Is JDS going to accept milk production data from a robot?" And it's because you can't really control how many times. Well, you can sort of control, but it's hard to control how many cow times per day that cow's going to visit the robot. What do you think?
Yes. You know, for us, the robot was in part because, you know, it's harder to find students who want to get up and milk cows. And were most of the work is done by students. And, you know, God love them. There's a lot of students who really want to, you know, they really like dairying, and they never had any exposure. They started on that farm, and then they wind up in a position in the field in dairy. I mean, we have several people like that. They love it. And you know, we're going to probably continue that, but it's harder to find students that want to get up in the morning and do that. So that some of it's that, and I feel like there's also, it is kind of the future. More and more are going in. There's some advantages of robots, and, you know, universities, if they're going to invest in a whole bunch of things, we need to all find some kind of way to be part of the future, right? Leading edge instead of the opposite end of that.
So. I think people are, I think that's where we're at. I always say, you know, it's just me, but I honestly think cows would rather deal with a robot. You know, you look at those big GIA robotic rotaries, you know, and those cows coming in the big robotic rotaries, and everything's the same. Every movement's the same. Every touch is the same. You know, same thing with the robotic feeders and the robotic pushers and everything else. The cows, they can understand that because they know what's coming at them, you know, and I think we're all going to end up there.
Most will adapt, and the few that won't, you know, then if in our case, we have, you know, farms in Worcester, and, you know, they'll just probably be up there.
They go to a different place.
So, you know, hopefully it'll work, but it's interesting, to find people to learn how to deal with that. It's a new system. And
Mhm.
as you know, in nutrition, it's harder to do. It's easy to do studies with a robot because
Mhm.
you can, feed them at the robot and you
Mhm.
can monitor it and so on. But it's hard to get intake of everything. So in JDS, we're still trying to figure out what to do with these kind of papers.
Yeah.
We know they're the future, but how do we get better control over full intake?
Yeah.
But we need to study how to use robots more efficiently. I think people who are interested in data science are going to find ways how to use the robot to get more information about individual cows that will be used. And we're going to have to do that too. So people will be into that.
So as we finish, we're going to finish now. And you got any specific areas of science and dairy nutrition that you see sort of on the horizon that, in your crystal ball, I think this is probably going to be somewhere where we either need to work on or something like that?
Well, there is, enteric methane. There's a lot of work being done on that, a lot of different kinds of additives and so on that and can have a role. I think we need to figure out when and where, better to help make them work more economically. So they're going to be more useful in the animals that are going to take advantage. So modes of action and so on need to be worked out. There's still a lot of that, trying to make sure that the rumen of every animal is as efficient as possible. There's people still studying differences among animals. How can one cow that's eating the same diet right next to another one have a different rumen microbial population? It's still,
how does that work?
It's not really totally different, but diet has an effect, but they do have some differences that are unique. So there's a lot of that. Amino acid nutrition is there's a lot of interest in that. If we can feed protein more efficiently, i.e. make sure they get the amino acids they need, either through microbial amino acids or through rumen integrated protein, we can feed less protein.
Right. Remember when we used to overfeed protein? What happened to those days? Gone.
20% protein.
Oh, wow. Yeah.
So now it's like, it's kind of like, how low can you go?
Right. Yeah.
And, but we need to find out how to do that on every farm. Like if you're going to go the lower in feed protein that you have, the more you have to be careful that you're actually feeding what you have. There's no safety factor left. So you have to do more sampling, feed sampling, and you have to have more management over cows and so on. So all those things come into play. I think there's a lot of people are studying fat and how to get more fat. Could be that we're going to try to manipulate the kind of fat. Maybe people want something different, and so on. So there's still lots of areas, and they're still drawing on the same fundamentals that drew me into, dairy nutrition in the first place. I started out ruminant nutrition, and the things I was interested in were like microbial protein. I felt like was more of an issue with cows, dairy cows. And those things are still coming in. We're still kind of sometimes going back to the old, the old glory days and figuring out how they did that. And we're adapting it with new techniques. So I think a lot of that is really cool, but boy, it's just hard to underestimate the power of technology and how we're going to use that in lots of ways, whether it's in the rumen or knowing when to feed products that we know work, just how to make them work more consistently. So a lot of that I think is the future.
I think I just like to say for you students out there, I hope we get some maybe some undergraduates that listen in, or grad students, and if you love cows, if you love chemistry, if you love feeds, if you love this sort of management, it all happened. It's true. I'm a repro guy, but it's the dairy nutrition sector. There's a home for you if you enjoy biology, chemistry, interactions of those kind, and just cattle and how they respond. There can't be anything more fascinating than, because there's just microbiology and physiology and I tell you, I'll just give a shout out. I mean, I tell you, Jeff, I ended up as a repro guy, and but I tell you what, if I had 2 lives to leave, I would love to take a shot at dairy nutrition. I think,
Would you?
yeah, for
Oh,
sure.
wow.
Oh yeah, for sure. Okay, but hey, we have a present for you, Jeff, and thanks for that. And you have to open it up on the mic, okay? Because
Oh, I do.
yeah, it's we promise it's not a snake that jumps out of the box. Okay, you open that very
Dairy digressions. Well,
there's not many of those mugs, my friend. There is
Well,
not
thank
many.
you.
Okay, so
yeah, well, that's really cool.
yeah. So anyway,
Thank you.
so this is it's been Dairy Digressions. We've been talking to Jeff Firkins from the Ohio State University. It's been a phenomenal discussion, and we're just so excited. So thank you very much. Thank you for talking to
It
us.
was great being here.
Thank you very much.